Good afternoon, sir. Okay. So good afternoon, one and all. So today's topic is secretory tissues. Okay. Is it visible? Screen and am I audible? Message me. Anyone? Okay. So today we will discuss about the next that one is the special type of tissues. Okay. So in last class we discussed about first one is the in tissue systems. Sorry, in the tissues. So different types of tissues are there. That is the meristematic tissues, and next one is the permanent tissues. So along with this, so another type of tissues are there. They are called as the special tissues or secretory tissues. Okay. So this is also important for competitive in view of competitive exams. So today we will discuss about this secretory tissues. So another name for this secretory tissues is the special tissues. Okay. So generally plants release many substances from their cell cytoplasm to the exterior. So they are called as the secretions. Okay. Plant secretions or excretions sometimes. Okay. So these are called as the, so these substances are released by some special type of cells. They are called as the secretory cells. Okay. So among these secretions, some are beneficial to the plant and some are not. So generally, the plants generally they are beneficial to the plants only, but some are not. So the beneficial substances which are secreted from the plant pods, they are called as the secretions. Okay. So the secretions may be water, nectar, salt, tannins, resins, latex, or gums. So when we can observe these gums from some plants, okay, and digestive enzymes and hormones, etc. So these secretions, it may be a water or it may be nectar, generally in the flowering plants. So generally we observe this nectar, salts from generally from the mangroves, mangrove trees, okay, and tannins, resins, latex, gummy leaf. From the gummy leaf plants, we can observe these gums, secretions or excretions. And the digestive enzymes, generally these are seen in the insectivorous plants and hormones, etc. So here secretory cells means, so the cells are tissues which facilitate that secretion. It is termed as a secretory cell or secretory tissue. Okay. So here again, these secretory tissues are again classified based on their position into two types. That is external secretory tissues and internal secretory tissues. So based on their position in the plant body. So they secrete externally or from internally. Okay. So based on their position, external secretory tissues and internal secretory tissues. So say here the secretory system in plants. So here secretory system, it is classified into external secretory system and internal. So examples for this external secretory tissues are glandular trichomes, nectaries, hydathodes. So this comes under the external. Okay, again here the nectar is again classified into floral and extrafloral. Whereas internal, that is again secretory cells, cavities and ducts, and latiferous tissues. So again, this one here, cavities and ducts again classified into lysogenous and based on the origin, cygogenous cavities. So again here, latiferous tissues are cells. Again they are non-articulated or articulated. Again articulated, anastomosing and non-anastomosing. Okay, they are connected. If they are connected, articulate, so they are called as anastomizing. So without joints, so they are called as a non-anastomizing. Okay, so this is the classification of secretory system, that is external or internal. So again, external glandular trichomes, nectaries, hydathodes, secretory cells, cavities, and latticevirus. So first one is the external secretory tissue. So the external secretory tissue, generally it occurs in the external surface of the plants. That is, so it may be unicellular or it may be multicellular, with or without the vascular element. So here these external secretory tissues, so it may be present in the external surface of the plants. And they may be unicellular or it may be multicellular. Okay, with or without the vascular elements. So they may contain vascular elements or it may be absent. So they are usually developed from the epidermis or subepidermal layers of the plant organ. So external secretory tissues which are developed external surface, that is on the epidermis. Okay, so they develop epidermis or subepidermal layers. So here the three main types of external secretory tissues are, first one is the glandular trichomes, nectaries, and hydathodes. So first one is the glandular trichomes. So generally these glandular trichomes, they are unicellular or it may be multicellular. So they are originated from the epidermis. So generally they are called as epidermal hairs. So they secrete some secretory, okay, cells. So usually they have a stalk and a head. So these trichomes, they have a stalk and a head. So the head portion will be secretory in nature. So they secrete some chemicals. And the heads of the glandular trichomes are covered with cuticle. And the secretion accumulates in the space between the cell wall and the cuticle. So the cuticle burst to release the secretions. So this is covered, and so when this cuticle is burst, the secretions will be released out. So they are called as the glandular trichomes. So examples, so these are the glandular trichomes, that is, they are originated from the epidermal region. So they contain stalk and head. Okay, so here in the head region, so these secretory substances are present. So it is covered with the cuticle. So when this cuticle is burst open, so the secretions will be released out. So this one is the first one is the glandular trichomes. So next one is the digestive glands of the Drosera. So generally digestive glands. So Drosera, it is an insectivorous plant and they capture and digest the small insects using the stalked mucilaginous glands. So they contains the tentacles. So with the help of these tentacles, they capture the insects which are present on their surface, and glands are glandular trichomes, and these glandular trichomes of Drosera possess vascular elements. That the tentacles, with the help of these tentacles, they capture the insects. Okay. So next one is the salt glands of mangroves. So salt glands are an external glandular structures which are present on the epidermis of the mangrove plant. So generally these mangrove plants, they are grow in saline water. So they are abundantly present in the leaves of halophytic plants, and they help to remove excess of organs. Okay. So generally they release excess of salt from the cells. Okay, so these are the example, that is the salt on the leaf surface of the black mangrove. So another example for glandular trichomes is the collectors. So these collectors are specialized external secretory structures which are found in some plants like malus, rose, and so they are found on the young leaf primordia. So generally these are present on the young leaf primordia, and these are the specialized external secretory structures. Okay, so they collectors, they produce a sticky fluid which covers the leaf primordia. So when we observe when we touch some plants, so when we touch the plant, so some sticky nature is observed. So this is due to this presence of these collectors. So the collectors drop off as a leaf primordia matures. So they protect the dormant bird and young leaves. So this the main function of these collectors is to protect the dormant bud and young leaves. So sometimes when we observe the plants, so some sticky, when we touch the plants, so some sticky nature is observed. So this is due to the presence of these collectors. These are the some secretory cells which are present on the external side. Okay. So generally these collectors, they are present at the young leaf primordia, and then the main function of these collectors is to protect the dormant bud and young leaves. Okay, so another one is the nectaries. Okay, so these are the examples for first one, that is the here. First secretory cells are classified into two types, external secretory tissues and internal. So in the external secretory tissues, first one is glandular trichomes. So these all comes under the examples of glandular trichomes. First one is the digestive glands of Drosera and salt glands of mangrove plants and collectors. So these three comes under the first one, that is the glandular trichome. So second one is the nectary. The nectary is secretory. So nectaries are external secretory structures, and the secretory is sugary fluid, which is called as a nectar. Okay, so nectar means it is a sugary fluid. So this nectar attracts the insects and helps in the pollination. So generally these nectaries are present in the flowers. Okay, so the nectaries are usually multicellular and can occur in the epidermis, hypodermis, or on the trichomes. So nectaries are associated with the vascular structures and particularly the phloem. So the nectaries have thick cuticle and nectar accumulates between the cuticle and the cell wall. So during the release of nectar, the cuticle ruptures and the secretion of the nectar will be released out. So two types of nectaries are present in the plants. That is the floral nectaries and extrafloral nectaries. Okay, so nectaries means these are also an example for external secretory structures. So nectaries mean they are the sugary fluid. So generally, the main function of these nectaries are they mainly helps in the pollination, because for the sugary substance, the insects or birds, they will attract towards these nectaries, and they mainly helps in the pollination. So the nectaries are usually multicellular. That is, they are made up of many cells and can occur in the epidermis. They are present epidermis, hypodermis, or on the trichomes. Okay. So generally these nectaries are associated with the vascular structures, mainly the phloem. So these nectaries have a thick cuticle, and this nectar, which is accumulated between the cuticle and the cell wall. So during the release of nectar, the cuticle will be ruptured and it is released out, and it lasts only for short time only. Again, here these nectaries are based on the presence. So generally we know that the nectaries are present in the flower. So they are called as the floral nectaries. So if they are present other than the flower, it is called as the extra floral. So floral nectaries, they are the common type of nectaries which are present in the flower. That is, they are associated with the flowers and usually found on the floral thalamus. That is the base of the gynoecium or androecium. So generally in the flower also they are present in the at the thalamus region. That is the base of the gynoecium. So where these gynoecium and androecium, that is, are attached. Okay, stamens and petal, stamens and carpel are attached in that region. So these nectaries are present. So other than these, extrafloral nectaries are seen. So these extrafloral nectaries, they are present on the vegetative parts such as the petiole, pedicel, or stem. Okay. So petiole means that is the stalk of the leaf or stalk of the flower or stem. So other than this, okay, so extrafloral nectaries are common in families such as the Euphorbiaceae, Verbinaceae, and Bignoniaceae. So extrafloral nectaries, they are seen in the mainly in the Euphorbiaceae members, Verbinaceae, and Bignoniaceae. So next one is another example for this external secretory tissues, that is the hydathodes. So hydathodes, these are a type of secretory tissues which are seen in the leaves. Okay. So they are modified pores, especially on the leaf, and which exudes water as a drops. So generally we observe in the grasses. So during winter season, so some what you call that is a guttation, the process of that is release of these water as in the droplets, so it is called as guttation. Generally, we observe on the grass leaves. Okay, so the pores of the hydrathodes are called as the water stomata. So generally, stomata that is they mainly helps in the transpiration and that is the release of air. But here water is released, so hence it is called as a water stomata. That is here they're releasing the water as a droplet. So they are facilitate the guttation. So secretion of the water as a droplet, and these hydathode, it is made up of group of living cells called as the epithelium with the numerous intercellular spaces. So the cells of the epithelium lack the chloroplast. So these vascular bundles of leaves end in the epithelium. So epithelium means, so these are the living cells. So it may, here the hydathode is made up of the epithelium, group of living cells, parenchymal cells without chloroplast. Okay. So epithelium cells open out into one or more subepidermal chambers. So this substomatal chamber communicate with the exterior through the pore. It is called as the water stoma. That is small opening which is covered with this epithelium. That is a group of cells. So, however, they are larger than the ordinary stomata. And these hydathodes always stay open since they do not have the opening and closing mechanism. So, like the in stomata, so here there is no opening and closing stomata. So, generally they are always open only. So, when the more amount of water is spent, they will be released out in the form of water droplets. So, that is the release of water in the form of water droplets from the leaf. It is called as a guttation. Okay. So, guttation. So, generally we observe these during the winter season, and the grasses mainly we observe these droplets on the leaf. Okay, so this is the structure of the hydathode. So it contains small opening and it is covered with the epithelium, and here the substomatal chamber, and here the vascular bundles are end here only, epithelium region only. So these three are the examples for external secretory tissue. So first one is the glandular trichomes, second one is the nectaries, and third one is the hydathodes. So they are present on outside, that is on the epidermal region, hence the name external. Okay. Next one is the internal secretory tissues. So internal secretory tissues. So internal secretory tissues, they are embedded inside the plants and they cannot be visualized externally. So they are present inside, hence the name internal secretory tissues. So they store the secretory products. So these internal secretory system of the plant may be single celled or it may be multicellular. So these internal secretory system, it is made up of only one cell. It is called as a unicellular secretory cells, or it may be multicellular. Okay, so in some plants, so it is highly specialized and complex secretory tissues may be present. So it is made up of more number of cells and some special system. So three types of internal secretory systems are seen in the plant. So first one is the internal secretory cells. Okay, another one is the secretory cavities and ducts, and third one is the laticifers. So here three types of secretory system, internal secretory systems are seen in the plant. So first one is the internal secretory cells. So they are made up of only one cell. Okay. So they are the specialized secretory cells with the secretions. So they are called as the idioblast. So another name for these secretory cells is idioblast. So secretory cells, they are found in the dispensed in the normal cells only, and the cells may be the idioblast, it may be isodiametric or elongated and tube-like. The shape may be, it may be isodiametric or it may be elongated or tube-like structure. And most of the idioblasts, they contain the secretory contents. So they can store tannins in these secretions. It may be a tannin or mucilage or oils or crystals like raphides and crystallolites. That is calcium oxalate crystals like that. So they can store. So here the secretory cells, they are made up of only one cell and the shape may be isodiametric or it may be elongated or tube-like. Okay. So they are present found along with these normal cells, but they store the secretory substance like tannins or mucilage or oils or crystals. Okay. So second one is the secretory cavities and ducts. So these are the cavities which are present inside the plant body with secretory contents. So based on their origin, again it is classified into two types, lysogenous cavity and cytogenous cavity. Okay. So lysogenous cavity means, so the lysogenous cavity is formed by the lysis of the some cells. So here the formation of this lysogenous cavity which is taken place due to the lysis, that is the death of some cells. So due to which some cavity is formed. So here in this cavity, so they secrete the substances. Okay, lysogenous cavity which is formed by the lysis of some cells which are filled with the secretory contents. So they are usually irregularly sized and shaped. The size of this lysogenous cavity is irregular. And generally it is found in the rutaceae members, citrus, lemon, etc. Next one is the cytogenous cavity. So the cytogenous cavity, these are formed by the separation of the cells by the breakdown of the middle lamella. So these so secretory cavities. Okay. So here the formation of cavity will take place in this. Here they secrete some substances. Okay. So here this first one is the lysogenous due to the lysis of the cells. Hence the name lysogenous. Here the formation of cavity which occurs due to the lysis of cells, and in this cavity they secrete the contents. Whereas in cytogenous they are formed by this cavity which is formed due to the separation of the cells by the breakdown of the middle lamella. So there is no cell lysis occurs. So these cavities more or less isodiametric or circular in cross section. And here the cytogenous cavities, they are internally lined with the interact secretory cells called as the epithelial cells. So in this cavity inside, here the epithelial cells are present. So these epithelial cells secrete the contents into the cavities. Okay, so examples for the secretory cavities. Generally, it is seen in the Ostraceae members are coniferales and Apiaceae. So here this is the lysogenous. Here the formation of lysogenous cavity due to the lysis. So here the cells are present. So due to the death of these cells, here the cavity is formed, and here they are secreting the substances. Whereas in cytogenous cavities, so they are formed due to the elongation of the cells and release. Leaving the some space. So here the formation of cavity will take place. So here the outer line which is inner line which is covered with the epithelial cells. So these epithelial cells, they secrete the substances and they are released into this cavity. This is the cytogenous cavity. Okay. Resin canals in the pinus. So another one is the third one, that is the laticiferous. Laticiferous tissue. So here it is fused. These laticifers, these are the fused tube-like cells which are filled with the fluid called as a latex. Okay, see, so these laticifers generally confined to the phloem, and however they can occur in the any part of the body. So generally it is confined to the phloem, but these latex, it may be secreted at any part of the body. So again, it is two types. Laticiferous in the plants. Again, they are non-articulated and articulated laticiferous. So first one is a non-articulated laticiferous. So these non-articulated laticiferous are called as the latex cells because they secrete the latex. So they are originated from the single cell and they occur in the intercellular space of the other cells. So non-articulated laticiferous, it may be branched or unbranched. So examples for unbranched non-articulated laticiferous are euphorbia and nerium. There is a branched non-articulated laticiferous are ficus. So here are non-articulated, that is they are made up of they are originated from the single cell and they are present in the intercellular spaces of the cells, other cells, and here they are not joined, but they may be branched or unbranched. So examples for unbranched non-articulated laticiferous are euphorbia and nerium. There is a branched non-articulated laticiferous are ficus. So articulate laticiferous, so they are also called as a compound laticiferous and they are originated from the series of cells. Here non-articulated, it is made up of from the single cell, but here the series articulated laticiferous, they are originated from the series of cells and these cells are arranged end to end. hence the articulated, that is jointed. So the common wall between the cells, it may be interact or disorganized. Okay, so the articulated lattice cells, they resemble the xylem vessels. Hence also known as the latex vessels. Here articulated lattice cells, another name is latex vessels because they looks like the xylem vessels. So there are again two types. Articulated anastomosing and articulate non-anastomosing. So here anastomosing means so they are connected. So joints are present and here the connection between the two cells will take place. It is called as the anastomosing. Non means they are not connected with the, if the two cells are present, they are joined, but inside there is no connection. It is called as a non-anastomosing. So here the latex tubes. So in articulate anastomosing latticefers, the lateral connections are established with the similar latticefers to form the reticulate structure. Example, hevia and money heart. Whereas the articulated non-anastomosing, so here articulate non-anastomosing lattice. So no, there is no lateral connections are established. So example is ipomia, musa and convolvulus. So hevia, so this is an example for hevia. It is the articulated anastomosing. So these are the examples. So next one is the cell structure of the latticifers. So latticifers cells, they contains a living protoplast and vacuoles are well developed and it stores the latex, and cells usually multinucleate, that is more number of nuclei are present in the cells, and latex may be milky white or colored or colorless. So here the color of the latex, it may be white color or it may be colored different colors or it may be colorless. So here the milky white latex, which is secreted by the euphorbia and hevia, and colorless latex by nerium, and orange or yellow latex by papaver. Okay, so next one is the chemical composition of the latex. So generally the plant latex, it is a mixture of many organic compounds and they contain the carbohydrates, organic acids, alkaloids, terpenes, resins and enzymes. So the latex of the papaver somnifer, mainly the latex of this papaver, they contains alkaloids like morphine and codeine. Whereas the latex of Carica papaya mainly contains digestive enzymes, that is papain. Okay. And latex of hevia, which contains rubber. So the rubber particles are suspended in the latex. So when the latex is removed from the plants, the rubber particles clump tougher. So this is about the secretory tissues. So generally, along with these meristematic tissues and permanent tissues, so cells also secrete some substances. So these are secreted by the special type of tissues. They are called as a secretory tissues. Okay, so these secretory tissues are again classified based on their origin into external secretory tissues and internal secretory tissues. So examples for external secretory tissues are glandular trichomes, nectaries, hydathodes. So glandular trichomes examples, again examples for this glandular trichomes are digestive glands, salt glands, generally they are seen in the mangrove plants, and collectors. So generally they are seen under Malus, Rosa and Pyrus. So the main function of these collectors is to protect the dormant buds and young leaves. Nectaries, generally they are the sugary fluids, so which may be present. So generally they are present in the floral nectaries. So along with this, so sometimes extra floral nectaries are also present. So that is they are present other than the floral parts like petiole, pedicel or stem. So hydathodes. So these are also known as the water stomata. So because they release the water in the form of droplets. So generally they are seen in the grasses. So these water stomata, the hydathodes, mainly helps in the guttation, that is the release of water. Okay. But these hydathodes are always open. Next one is the internal secretory tissue. So here internal means they are present inside the plant and they can secrete the some secretions. So again, these are internal secretory cells, secretory cavities and ducts, and lattice cells. So secretory cells, they are made up of single cells. Okay. And mainly the another name for the secretory cells is the idioblast, and they contain some secretory substances like tannins, mucilage oils, or crystals of salt, sodium carbonates, or sodium crystals. Okay. Secretory cavities, so they are made up of a group of cells. So again, they are lysogenous cavities, and based on the origin, lysogenous cavity, cytogenous. So if the cavity which is formed by the lysis of the cells, it is called as the lysogenous. Whereas the cytogenous, it is formed by the, that is the separation of the cells and leave the some space. So it is formed the cytogenous cavity. So another one is a lattice phase, that is the laticeferous tissue. So these are the fused tube-like cells which filled with the fluid called as a latex. Okay, again it is two types, non-articulated and articulated. So non-articulated means these are the latex cells. So they originated from the single cell. Okay, and here again these non-articulated lattice phase again classified into branched and unbranched. So example for unbranched is euphorbia and anerium. There is a branched non-articulated lattice phase is the ficus. Articulated lattice phase, so they are made up of the group of cells, and hence it is called as articulated, that is a compound lattice phase. So the common wall between the cells may be intact or disorganized. And here, again, this is articulated anastomizing and articulated non-anastomizing. So articulated non-anastomizing means, so the articulate, they are lateral, if the lateral connections are present between this lattice phase, it is to form the reticulate structure. So it is the articulate anastomizing. There is a articulate non-anastomizing means, so there is a no lateral connections. Okay, so example for this articulated anastomizing is Hevia and Manihat, whereas articulate non-anastomizing is Ipomia and Musa convolvulus. So generally, so the latex based on this color of the, so generally these latex cells, these are the latticeferous cells are living cells. They contain a living protoplast and vacuoles also well developed. And the mainly they store the latex. Okay, so the latex, it may be a white color or colored or colorless. So white latex is secreted by the euphorbia and hevia, and colorless latex is seen in the nerium, and orange or yellow latex is seen in the papaver. So chemical composition, generally the latex which contains different substances like alkaloids, terpenes, or resins and enzymes. So the papaver somniferum, they mainly contains alkaloids like morphine and codeine. So these points are important. And carica papaya, they contains the digestive enzymes papain. So papain is seen in carica papaya like that. So these are important points. And latex of hevia, they contains the rubber. So and rubber particles are suspended in the latex. And when the latex is removed from the plant, the rubber particles will be clumped together. So this is about the secretory tissues. Okay. So today I will stop here. In next class, we will discuss about the tissue systems. Any doubts? Okay, so I will stop here. In next class, we will continue. Any doubts? Message me in the chat box if you have any doubts. No doubts? Okay. No doubts? Okay, I will stop here. Okay. Okay, girls. Bye. Okay.
Madam, shall we going to wind up? Hello? Audio, sir. మన సార్ లోపల అక్కడనే ఉన్నాడు. మన సార్ అక్కడ ఉన్నాడు. మన సార్ లోపల ఉన్నాడు.